Hello, ladies and gentlemen. My name is Tanika Jackson, and the title of my project is Genetic Variation versus Environmental Contaminants in Striped Bass in the Anacostia River. With my project, I attempted to analyze single nucleotide differences in 18 striped bass fish samples for mutations due to endocrine-disrupting chemicals. My thesis is excessive runoff water that flows into the Anacostia River containing pesticides, herbicides, toxins, and trash can have detrimental effects on the river's health, which directly translates to my hypothesis, which is there is a correlation between endocrine disrupting chemicals or environmental contaminants, genetic variation, and striped bass. This picture that you see in front of you is a catfish, and while it is not the fish that I researched, it is one of the species that is found in the Anacostia River. The pink thing that's on the tip of its face is a tumor, which scientists have concluded are caused by environmental stresses, and specifically in the case of the Anacostia River is due to poor water quality. One type of pollution that I found in my research that contributed the most to the river's poor water quality is chemical pollution, or specifically endocrine disrupting chemicals, which find their way into the river through runoff water. And also in my research, I found that 75 to 90% of the pollution in the Anacostia River is due to runoff water pollution. And endocrine disrupting chemicals are any chemical that has the ability to disrupt the chemical pathway or the chemical signals of the endocrine system, which controls the reproductive system or controls the hormones of the reproductive system, like testosterone and estrogen. And estrogen was the main hormone that I had researched. So that is how I planned my experiment, which I did through an internship at Georgetown University. If these endocrine disrupting chemicals are mutating the fish, there should be a difference in the DNA, and while it may not manifest itself physically, it could manifest itself genetically. For this experiment, I used PCR, or polymerase chain reaction, on 18 fish samples for the gene aromatase B through primer design and electrophoresis. Aromatase B is an enzyme that converts estrogens into androgens. Actually, it converts androgens into estrogens, and it's prevalent throughout a fish's life. It actually doesn't go backwards. It's only androgens into estrogens. Excuse me for that. I chose this gene because of its relationship to the reproductive system of the fish, and also I chose this gene knowing how endocrine disrupting chemicals affect the reproductive system. And there are two types of aromatase. As you can see, it's aromatase A and there's aromatase B. Aromatase A is usually found in like the gonads of a fish, and aromatase B is found in the hypothalamus. And I chose aromatase B because of its location, and the fact that it's prevalent with the fish's life was a big factor as well, because aromatase is not prevalent throughout every organism's life, such as in humans. It stops at infancy, which I thought was pretty cool. So I was able to work with this gene through PCR, or polymerase chain reaction, which is a tool used to make millions of copies of a specific gene or locus in the genome. This picture that you see here is a diagram of the process. The key to PCR is primers, which are the yellow tags in the diagram that you can see. Without these, there is no way to find the specific part of the genome that you were looking for. My primers were custom designed, which took quite some time, 4 months actually, and it took so long because striped bass have not been sequenced before, and without a DNA template, it is difficult to synthesize primers, but I did. So these are my final primer designs, and this is really a small fraction of what I did. I must have done at least 20 primer designs before I narrowed it down to these 4. And on the right hand side, those are my conditions for my primers. And the second one that I used was the one that worked the best, and it wasn't completely successful, but it did amplify some product, which you can see here. These are my gels that I ran, or my gel electrophoresis that I ran after to see what kind of product I had. The gene itself is about 1,200 base pairs, so it's a really large product, which is why it's really high up on the BP scale. But in the first gel that I ran, you can see I really only had one product out of like the 18 that I ran, and it was ambiguous, so I had to do it again, which is the second one, which is super light because of all the ethidium bromide that I used. But it did have a successful amplification. And the second step of my project was to do DNA sequencing, but the third gel that I ran was completely unsuccessful, which gave me inconclusive results. So, in reflection, I did have inconclusive results, but if I had time to do this again, I would have a broader range of genes and more species of fish. And I would want this to be a new way to monitor water quality. Thank you so much for listening. I hope that my project was interesting, and I hope you are as passionate now about water quality as I am. Thank you.